PGT stands for Preimplantation genetic testing. It is a specialized procedure used in conjunction with in vitro fertilization, or IVF, to test embryos for genetic conditions or chromosomal abnormalities before implantation into the uterus. This helps ensure that only healthy embryos are selected for transfer, increasing the chances of a successful pregnancy and reducing the risk of genetic disorders in the baby. PGT is divided into three main types based on the purpose of testing. First, PGT-A, which stands for aneuploidy. Its purpose is to detect chromosomal aneuploidies, meaning an abnormal number of chromosomes. Examples include Down syndrome, which is trisomy 21, and Turner syndrome, which is monosomy X. PGT-A is often recommended for women over age 35. It's also recommended for those experiencing recurrent miscarriages, repeated IVF failure, or male factor infertility. Second, PGT-M for monogenic disorders. This tests for single gene disorders that are inherited, such as cystic fibrosis, sickle cell anemia, Tay-Sachs disease, and Huntington's disease. PGT-M is for couples who are carriers of specific genetic disorders or families with known genetic diseases. Third, PGT-SR tests for structural rearrangements like translocations and inversions. The PGT process begins with an IVF cycle. First, ovarian stimulation and egg retrieval take place. Next, eggs are fertilized with sperm in the laboratory to form embryos, which are then cultured to the blastocyst stage on day 5 to 6. During the biopsy stage, a few cells, typically 3 to 5, are safely removed from the trophectoderm, which is the outer layer of the embryo. The embryo is then frozen afterward. The biopsied cells are analyzed using advanced techniques such as next-generation sequencing, array comparative genomic hybridization, or PCR for single gene disorders. Finally, genetically normal embryos, called euploid embryos, are selected for frozen embryo transfer. The benefits of PGT include reducing the risk of genetic disorders, increasing IVF success rates, and decreasing the risk of miscarriage. It also avoids the ethical and emotional burden of termination after prenatal testing and can help with sex selection when legal and ethical. However, PGT has limitations. It does not guarantee a healthy baby, as it only screens for known or detectable conditions. The biopsy may slightly affect embryo viability, though this risk is very low with modern methods. PGT is costly and may not be covered by insurance, and it requires IVF even if a couple is fertile. Suitable candidates for PGT include couples with a family history of genetic diseases, those experiencing recurrent pregnancy loss, women of advanced maternal age, or those with previous children with genetic disorders. Also suitable are those with recurrent IVF failures or known chromosomal abnormalities in one or both partners. The most common technique today is next-generation sequencing, or NGS. It detects aneuploidy, single gene disorders, and structural rearrangements with high resolution and accuracy. Polymerase chain reaction, or PCR, is used in PGT-M for single gene disorders. It amplifies DNA to detect mutations in specific genes, but requires knowing the specific mutation in advance. Modern PGT uses day 5 to 6 blastocyst stage biopsy, which targets the trophectoderm cells that form the placenta. This is safer for the inner cell mass that becomes the future baby. This approach is more accurate and is currently the standard of care, offering hope to couples seeking to build healthy families through advanced genetic testing.